Welcome all of you. In this session, we are going to see about the soil fertility evaluation. So in the previous videos, we have seen the first two methods, that is visual method of soil fertility evaluation and analysis of plant, the whole plant as well as the tissue testing. These two methods, how the soil fertility can be determined, that we have seen. And in this session, we are going to see about the biological test with the higher plants and certain microorganisms. So in this soil fertility evaluation, certain higher plant as well as microorganisms are utilized. How they are involving in a soil fertility analysis, we will see. In biological test, the first one is use of higher plants. Under this, several fertilizer trials, field plot method will be conducted. The first method is simple fertilizer trial, SFT. So this research will be conducted in research station, and it is not applicable to the farmer field. And this SFT gives idea about the hidden hunger. Hidden hunger already we have seen. The plant will not show any deficiency symptoms, but it needs more amount of nutrient. And usually the effect of macronutrients are tested, such as NPK. So among these NPK, different combinations will be created. So here you can see 8 treatments. Like first one is control: nitrogen, phosphorus, potassium, nitrogen plus phosphorus, nitrogen plus potassium, phosphorus plus potassium, and finally all three nutrient combination. So using this simple fertilizer techniques, we can find out which plot, different plot we can prepare in this plant. For example, this is nitrogen plus phosphorus. This is phosphorus plus potassium, and this is potassium plus nitrogen. Different plot, different treatments will be received. So using this, which plot giving more amount of yield or more productivity, based on that how the soil performance was there, this will be assessed. Second, complex field trial. So along with the macronutrient, micronutrient also included. Many interaction studies are possible here. So the recommendation can be extrapolated to other soils with a similar characteristics. For example, if you see the insecticide will spread all over the country. If you are giving the same treatment to the insecticide, the same will be given to the another place. Okay, so this is what the complex field trial is happening. The next trial is a strip test on farmer field. So here narrow field strip will be created with the selected nutrient treatments for individual strips. Okay. This will be used for verifying the accuracy of recommendation, how the accuracy of nutrient is producing the yield or growth of the plant. So this treatment is in similarly uniform areas as possible, not ununiform area. The uniform area, you can use these treatments if several soil types or condition occurs, each soil type equally in each treatment. Okay, so if the soil types are differ and conditions are differ, each type of soil occurs equally in each treatment. So yield monitoring is needed very frequently. So it is very, very important in strip test on farmer field. This test can be conducted in farmer's field alone. And next test is laboratory and greenhouse test, where this is the simply and more rapid techniques utilize small amount of soil and to quantify nutrient availability, as well as the area requirement also. So very less, usually you will do this experiment in pot culture. So these soils are collected to represent wide range of soil properties. Specific nutrient, individual nutrient contribution will be evaluated, and sensitive element to the crops also we can analyze. For example, sorghum for iron, the iron role, the how the iron producing the growth of plant in sorghum can be identified. So this test reports iron deficient and iron sufficient soil, how much iron contributed, and how much iron is needed further for the upcoming treatment in the particular soil, and the decreasing iron deficiency with increasing DTPA extractable iron. So this iron chelated ions, this also can be used to decrease the iron deficiency. So these are all the trial we can conduct for soil fertility evaluation. Apart from that, in a laboratory condition, you can grow some certain crops. They classified into different, like Newber test. First one is, so in this Newber test, 100 gram of air dried soil plus 50 gram of coarse sand, 250 gram of fine sand you will be taking uh, in a container. In those samples means the soil samples will be moistened with water, then a large number of seedlings can be grown here. Nearly 100 rice seeds can grown and allow it to grow for 17 days continuously. By meantime, it will absorb all the nutrient from the soil, and the nutrient almost exhausted. And these seedlings finally removed and dried and analyzed for the nutrient uptake. So at the same time, you have to run a blank without soil. So from that, you can understand the difference between with the soil and the without soil, how the plant performance is. Okay. So finally, when you are deducting this value from this value, you will get the exact amount of nutrient concentration contributed from the soil. The second method is mutually spot culture method. So oats and cereal, oats or cereal will be grown in a pot containing 2.72 kg of soil. Nearly 2.5 to 3 kg of soil will be taken here, and 10 treatment will be allotted with the combination of NPK. Here you can see no nitrogen, nitrogen plus phosphorus, nitrogen plus potassium, and NPK combination together. So these are all comprises almost 10 treatments. So yield increased by the each factor or each nutrient will be calculated here using this formula. This is Mitchell's formula. You can see this is increment in growth. Dy is increment in growth. X is a growth factor. Because of this growth factor, how much yield is increasing. And C is a constant, and A is maximum yield, and Y is estimated yield. So by using this formula, you can easily get the soil fertility level using growing of oats or cereal in a pot culture. So from the NPK, these three pots NPK, the all three combinations is considered as a maximum yield, and the yield obtained in these three pots and these three pots are converted to percentage. When you are deducting the value from the highest to lowest, you will get some difference. Based on the table arrived from this value, the soil fertility level can be deducted. Jenny's pot culture method, the pot containing 1.81 kg of soil, so where Roman lettuce can be grown for 6 weeks continuously. So the treatments you can see, nitrogen 0, phosphorus 0, potassium 0 is like a control, and nitrogen 150, phosphorus 150, potassium 100. Okay, in this recommended dose, in this dose you can apply, and no nitrogen means your phosphorus and potassium will be applied. No phosphorus means nitrogen and potassium will be applied. No potassium, nitrogen and phosphorus will be applied. So this combination with 4 replication can be created, and finally the percentage values are grouped in 3 categories, and the table was arrived like this. In this yield you can see, based on the percent yield contributing from the nutrient nitrogen, phosphorus, potassium, sulfur, lime. So nitrogen, if the yield shows only 20 kg, 20 percentage, definitely there is a deficiency symptoms, deficiency of nitrogen. 20 to 50, there are some probability of nitrogen deficiency, and 51 to 70, there is no deficiency. Similarly for phosphorus, potassium, they have given a range based on the percentage yield obtained. Next technique is Stanford and demand technique, where this method is Newber method, modified Newber method. Means where in Newber method we have seen 100 dry seedlings were grown in the soil. So similarly here also the uh, crop can be grown in the soil containing 200 gram plus fertilizer, 200 gram plus fertilizer containing media. You can allow the crop for growing 2 to 4 weeks. So then after that you will remove the samples and analyze for their deficiency, moderately deficiency, and no deficiency. Okay. Then sunflower pot culture techniques for boron. This is very important. In this pot culture experiment, in the pot you will add all nutrient solution except boron, and you will allow 5 seedlings to grow. 5 seedlings of sunflower you will grow. So sunflower will vigorously extract the boron present in the soil. Just in the 0.5 kg of soil only you will be using here. So in this 500 gram of soil, if the nutrient deficiency of boron occur within 25 days, definitely deficiency is there. If 28 to 36 days, moderate deficiency. If it is more than 36, if it is more than 36, there is no deficiency of boron is observed here. Then use of indicator plants. Some plants are suited show the deficiency of nutrient, so they will show definite deficiency symptoms, exactly like nitrogen deficiency symptoms can be observed clearly from cauliflower and the cabbage. So it will give a clue about the deficiency symptoms from this particular plant. So we all know the chlorosis is a nitrogen deficiency symptoms that will appear very clearly from the cauliflower, cabbage, maize. Likewise, phosphorus deficiency will appear very clearly from rapeseed, potassium, potato. So these plants immediately show the deficiency symptoms if the nutrient are deficient in the particular soil. So for all the elements are having their indicator plant, you can see potassium potato, calcium cauliflower cabbage, magnesium potato, iron cauliflower cabbage potato, sugar beet, so for sodium sugar beet, manganese sugar beet oats potato, boron sugar beet. So this is called indicator plants where you can find out. Then coming to use of microorganism, there are different techniques using different microorganism. First one is Sackett and Stewart technique. So this method is mainly for identifying the phosphorus and potassium deficiency in soil using a acidobacter as a microorganism. So here the procedure for this microbiological method through Sackett-Stewart's technique is 3 containers will be having a soil culture. One is with supplied with phosphorus, another one is supplied with the potassium, and the third container will be both like phosphorus plus potassium. So this culture will be inoculated with acidobacter and incubated for 72 hours for allowing the growth of colony of acidobacter. So based on the amount of colony growth, the soil is rated deficient or sufficient or moderate. See here, there are 4 classes. The class 1 is very deficient, where none or few to numerous extremely small pinpoint colonies. The growth is affected here because of the deficiency symptoms. In class 2, moderate deficiency, few numerous but small weak colonies you can observe here. Okay, some weak colonies you will observe like this. And class 3, slight deficiencies, colonies on unfertilized and fertilized colonies on unfertilized plaques are equal in number and development. So some well growth can be seen here. And finally, class 4, there is no deficient, no evidence of deficient, where colonies on both fertilized and unfertilized plaques are equal. A good amount of growth can be observed because here no deficiency of nutrient. Second test is Cunningham plaque test. So in this test is mainly employed for identifying the phosphorus deficiency because this is very sensitive to phosphorus. So the soil is mixed with nutrient solution and the paste slurry is prepared and spread on a clay dish. In the clay dish, the Cunningham will be inoculated and it is allowed for growing in 4.5 days. So finally, after 4.5 days, you can see the growth of mycelial which will indicate the deficiency. If the mycelial growth having less than 10 mm in diameter, it is very deficient in phosphorus, and 11 to 15 moderately deficient. So when the diameter is increasing, there is no deficiency. Like more than 22 mm in diameter, no deficiency of phosphorus. Third test is Aspergillus niger test. So this test is mainly used for potassium content in soil to identify the fertility level of potassium. And the soil is taken in conical flask and nutrient solution is being added and inoculated with Aspergillus niger for 4 days. It is allowed for growing. Then weight of the mycelial pod and potassium content are taken into account. If the weight of the pod is less than 1.4 gram and potassium absorbed by 100 gram of soil is less than 15 milligram, it is very deficient. Similarly, you can see there is no deficiency when the pot weight is more than 2 and uh, 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 more than 20 milligram of potassium absorbed by the 100 gram of soil. Likewise, moderate to slight deficient showing 15 to 20. 15 to 20 here 1.4 to 2 gram of pod. The next test is Mulder test for copper and magnesium. Here the same Aspergillus niger is using. They will be indicating copper and magnesium deficiency in the soil. So known standard are prepared in weighed quantity of soil based on copper and magnesium. So after that, the Aspergillus niger is inoculated in the soil. Then the after growing, the color of the mycelia will be compared here, both in the treated soil and untreated soil or unknown soil. So if it is very deficient, the copper will be having less than zero point 0.4 microgram. Similarly, if it is having 1 to 1.4 microgram, slightly deficient. More than 2, there is no deficiency. Likewise, magnesium less than 50 milligram per 3 gram of air dry soil and 50 to 100 milligram more than 100 milligram. This classification is based on the milligram of magnesium per 300 gram of soil. Then this is the CO2 evaluation method where also you can find out the microorganism activity so that it is based on the carbon dioxide evaluation from the soil. So the soil will be incubated in a conical flask with the compost or super and potash for 10 days and the CO2 evaluation is assessed by absorption of standard barium hydroxide. In a conical flask, you will take the soil with the compost or fertilizer. Then you will insert the barium hydroxide solution in a vial. And when you are closing this, the carbon dioxide evolved from the fertilizer or from the soil will be absorbed in this barium hydroxide. So the excess barium, after absorption of carbon dioxide, the excess barium hydroxide is treated with, titrated with hydrochloric acid so that we will get a value. Based on the value, we will find out the carbon dioxide evaluation from the microbial activity, because it is the indirect measurement, like microorganism is respirating and releasing carbon dioxide, thus carbon dioxide is we are observing here. So the carbon dioxide evaluation can be identified here. If the carbon dioxide uh, uh, evaluation is happened in an increased manner so that the deficient nutrient is being added. That is the reason for increasing carbon dioxide evaluation. Thank you for attending the class. Thank you.